Yes, sir. Okay, we will start, sir.
Yes, yes.
Esteemed resource person of the day, Dr. Satish Kumar, Professor of Biotechnology, Bharatiya University. Respected Head of the Department of Botany, Dr. S.R. Sudhir Kumar, Dr. David Raja, and Dr. Johnny Kumar Thakur, the co-organizers of this webinar, and my dear participants, a pleasant evening to one and all. So it gives me a great delight to welcome you all to this third day session of International Webinar Series on Medicinal Plants Research and Drug Discovery. So dear participants, we appreciate you all for joining with us in this evening. So let us begin this webinar with God's blessing through prayer. Almighty and ever-loving God, we come to you today asking for your guidance, wisdom, and support. As we begin this webinar, help us to engage in meaningful discussion. Fill us your grace and strength. We ask you these things in the holy name. Amen. Thank you all. So it's a time to welcome our resource person and all our dignitaries. It's a great pleasure to welcome our resource person, Dr. R. Satish Kumar, Professor of Biotechnology, Bharatiya University. So we are very much privileged to have you, sir, to this great webinar. So I welcome on my own behalf and the Department of Botany behalf and the college management behalf, welcome you, sir. Hearty welcome to our resource person.
Thank you.
Welcome,
Thank
sir. A man with a great ideology, determinism, vision. He is our beloved Head of the Department of Botany, Dr. S.R. Sendil Kumar. Sir, we hope under your leadership and guidance, may our department may reach the next level of glory. With a high level of gratitude, I welcome our Head of the Department of Botany, Dr. S.R. Sendil Kumar. Welcome, sir.
Thank you, Francis. Thank you.
I am happy to welcome my dear friend, one of the organizers of this webinar, Dr. David Raja. He is always supporting me to organize this kind of webinar. At this juncture, I welcome Dr. David Raja to this webinar series. Welcome, Dr. Sir.
Thank you, sir. Thank you.
Yeah. I also welcome Dr. Johnny Kumar Thakur. Somehow he is supporting me to run this program successfully. I welcome Dr. Johnny Kumar Thakur. Welcome, sir. Last but not least, our participants. So whenever we are sending a circular to the participants, they are always acknowledging us and they are coming in good number. So at this juncture, I welcome our participants to this third day session of International Webinar Series. Welcome all participants. Thank you all. So it's a great pleasure and it is my duty to welcome and introduce our resource person, Dr. Satish Kumar. Dr. Satish Kumar is a professor of biotechnology at Bharatiya University, Coimbatore. He did his doctoral degree at the School of Biological Sciences, Madurai Kamara University. During his PhD, he awarded DAD short-term fellowships to work at Max Planck Institute for Molecular Plant Physiology, Germany. Later, he joined as a postdoctoral research in the Department of Botany, University of Hong Kong, where he gained expertise in plant molecular biology and genetic engineering. He joined in the Department of Biotechnology at Bharatiyar University in the year 2005 with an expertise in plant molecular biology and genetic engineering. At Bharatiyar University, he established the Plant Genetic Engineering Laboratory in the Department of Biotechnology. His research focuses on metabolic engineering, molecular farming, and DNA barcoding. Dr. Satish Kumar, he is a recipient of several awards and honors. Recently, he has been nominated as a fellow of Academy of Sciences at Chennai. He has received several research grants from various funding agencies like DBT, DST, DRDO, UKIRI to the tune of 3.5 crores. Apart from these, he is also a consultant for various industries like the Himalaya Drug Company, Green Chem, Arjuna Extracts, Lila Herbs, and Indus Biotech in the aspects of DNA barcoding. He was awarded full travel bursary continuously 3 times to deliver an invited talk in international conferences on barcode of life in Mexico, China, and in Canada also. Recently, he has been nominated as a subcommittee member of the panel on DNA barcoding constituted by Indian Pharmacopoeia Commission, Government of India. Currently, he is a member for Project Monitoring Subject Expert Committee constituted by Tamil Nadu Higher Education Department. He has delivered many invited talks in various institutions in India and in abroad. To his credit, he has one US patent and he filed one Indian patent. He has published one book, 83 publications in well-reputed journals with high impact factors. He has also published 23 edited book chapters and he submitted 400 DNA sequence in NCBI. Till date, he has guided 15 doctoral scholars and 20 MPhil scholars under his guidance. Currently, his team includes 3 postdoctoral fellows and 5 doctoral fellows. So in his credit, research credit, he has a Google Scholar Index 22. His Google Scholar Index is 22 with a citation of 1,690 citations. Recently, he has become a program coordinator for the new DST-sponsored MSc Medical Biotechnology course. Such an eminent professor and researchers with us. So I welcome him on behalf of the St. Joseph's College Management, on my own behalf, and the Department of Botany behalf, finally the participants behalf. I welcome you, sir. Welcome Dr. Satish Kumar, today's resource person. Please, sir, over to our resource person. Yeah.
Thank you for your kind introduction. Good evening, all. First, I would like to thank the organizers and co-organizer, Dr. Francis. And I am very happy to be here because it's not something first time. I've been there to St. Joseph's a couple of times for delivering lectures, but because of today's situation, still the activity is going on. It is good to know that in St. Joseph's things are going normal as usual. So I would like to thank the department, head of the department, and also the management, St. Joseph's College, for this opportunity. So I think it's a very different kind of situation where we are in. And I think when Dr. Francis called me almost like 3-4 weeks back regarding this webinar, we all thought it will be whole day, will be free and we can have any time. But suddenly the classes have started. So he was requesting whether we can change time and we can go ahead with the same topic in the evening. So I'm sure here also we have started. I just came back from the department. I thought it would be better to reach home and then start this. So I'm at home now. And I'm sure the students also would have attended the class and little bit tired of that online classes, but I will make it very short and sweet as much as possible. Interactive, of course, it is not possible suddenly. I mean, in between. We'll have it in the end. I think Dr. Francis said he will moderate it with the questions and other things. So what I'll do is, I don't have many slides. I'll restrict maybe 30 or 40 minutes maximum. But after that, we'll have enough time to interact as much as possible. Okay, I just want to know from Dr. Francis, what kind of audiences mostly we are having today? No, you are to unmute, sir.
We have participants from different parts of our country.
Okay.
And other countries also, from students, of course, young faculty members and researchers also.
So you could advertise, such a wide places.
So in every webinar, in our webinar, so more than 1,400 participants are participating.
Amazing numbers.
That is why
Right
we
now, it is going in YouTube, is it?
Yeah, right.
Okay, it is quite good. So I think looks like it's a big mixture of audience. So what I'll do is I'll cover the basic wherever a little bit with the details. You don't have to worry too much about it. I'll just brush through it. Anytime anybody can write to me because there is an email ID here. You can contact me for some kind of help in the lab or even theoretically or any help like whatever it is. So don't worry too much about different things which I'm going to talk. But basic idea is what exactly is DNA barcoding, and that's why I kept the topic very simple. The first question, whatever we talk, we have to ask what is that. Okay. The next thing what we will ask is how to do that, how easy it is, and finally why we need all this. Okay. So these are the things which I'm going to answer in this very short time. Okay, so DNA barcoding as such, I think it's not something new for me at least, because we are working from almost 10 years on this topic, more than that, and 2 or 3 students have completed PhD. So we started just like any other lab, from the very basic, doing directly the barcoding, but now we have slightly shifted, and we are using it for different applications aspect. Okay, which I'll be talking in the end. So first we'll see what exactly is DNA barcoding. In the slide itself, you can see on the right hand side, there are some lines like a barcode, which we see normally in the supermarkets in order to find the product or price of the product or any information in the on the product. So similarly, whatever we want to know about the species based on the nucleic acid sequence, we can make it as a barcode, and then using a scanner, we can quickly identify what is the species or know about the species. So that is the reason why the same barcode, whichever we use commercially in the supermarket, here instead of the product, information, here we are using nucleic acid as information. That's why it is called as DNA barcoding. So as simple as that. And it has become something like a very trending topic now, because it has found so many different applications currently. So it has become not only as for naming the species or identifying the species. More than that, it has become more of commercial use, like authentication and other things. Let's see all that one by one. So the question is, what exactly is DNA barcoding, and why do we need that, or why do we need suddenly one new technique when we have very conventional taxonomy, especially in India, where we have so many good taxonomies, and why do we need this tool? But the question now is, before answering that, the big question now is, no one knows how many species are there in the universe. So this is a big question, even after all these advancements. So that means all the biodiversity resources, especially country like ours, tropical country, I think we are all cherishing whatever the diversity which we have, which normally doesn't occur in the temperate countries. So these are all some of the big treasures we have for India like country. So that means we have, we have to give more importance to know about that, so that we can conserve it, so that we don't lose it, and future generation also should get benefit of it. Okay, so we have to answer first, what exactly, how many species are there in the universe? If you see here, there are some statistics which I can show from the literature, but even this, you can see from the beginning, or whatever the taxonomists could do all these years, you can see the numbers. It is not a big numbers what we have right now, but it doesn't mean that taxonomists have not done anything, but they've explained the species, and they've given all the taxonomical aspects of that particular species. So that's why we could use all this today. I mean, we could use for research or any other purpose of taxonomy. because of taxonomy. Okay, so these are all some of the statistics which we have. Whatever the taxonomist has explained very thoroughly in all the groups from plants, invertebrates, vertebrates, and all this. This accounts for 1.5 million to 2 million described species. So till now, from the beginning, taxonomists could explain 1.5 million to 2 million described species. So this number, they say, roughly, it is not even half of the number, what exactly the real numbers which they feel it will be there in the universe. That's why in the newspapers or any of the magazines or in the news item, suddenly we see one institute has discovered new species, or some institutes have discovered new species and named, maybe it's the first time in the world. Like that, it is going on all over the world. So that means there are many more things have to be explored, and there is a need. We need to do that fast. The reason being, 1/3 of all the species, they are saying it is getting threatened, or we all know the reason, because of urbanization, many other things, because of industry and all that. Occupying all the lands and other things. So 1/3 of all the species are threatened. I'm talking about the whole species which we are taking into account. So that means we need to act fast before we lose many of these things, before even we identify that. So that means we need some kind of a high throughput technology in order to at least not exactly like taxonomic kind of explanation which we can give, but at least we can give some kind of ID to the species and then put it in the database, so that later somebody can be using a taxonomist, or somebody can slowly sit and describe that. So basically, we need a technique in order to do these kind of things, that is to identify or to name the species as an ID, and then at least we can start conserving it so that we don't lose of all these things for future generations. So this is one area which I want to emphasize before we go into the topic. Then we can realize how important the DNA barcoding is. Okay, this is another very nice slide to represent how much need is there in order to use some kind of a high throughput technology in order to explore the biodiversity, especially country like ours where we have so much of biodiversity. On the left hand side, you see the screen. There are very small squares with the different colors. Different color means different groupings. So one small square is equal to almost 10,000 species. So whatever we have explained in the previous slide, 1.5 million to 2 million, has been split up here and then arranged as a small square. So if you see on the left hand side, these are all the discovered species, fully explained species by the taxonomist, and which accounts you can see here, 1.7 million, they say on the left. If you fix that in the total number of species in the universe, you can see on the right, it accounts only for maybe 1/4 or 1/5 of the big square. So that means we have to fill those blank squares as quickly as possible before it gets extinct or disappear. Okay, so you can see here almost like 10 million species, they say, and in that we have just covered 1/5 of it. So that means we need to do something in order to explore rest 4/5 of the new species which we have. Okay, so all these are statistics just to give the importance. Even though all these advancements which is happening, still we are yet to to name or to give ID for all the species in the universe. Okay, so still we are long way to go. Okay, so when people were thinking about all this, then there was a publication, I think in 2000. Okay, so if you see this, it's a very, very prestigious, we can't say it is a fully journal or something. It's a magazine, science magazine, very prestigious magazine. Suddenly there was a cover page article. You can see their barcode, and also they give an analogy of zebra. So basically what it means is, take the DNA of that particular species, and then you sequence it, and then put the sequence as a barcode, so that that barcode can be used to name or name a particular species. Okay, so this term was introduced in 2000, year 2000, and they named it as DNA barcoding. As I told you in the beginning, barcoding is nothing but a barcode which we use to know any information about the product. Similarly, here we use DNA, so it is called as DNA barcoding. Okay, so it is direct meaning of that. Okay, when it came, it was described as it's a new diagnostics tool for rapid species recognition, identification, and speed discovery. So in the sense, we can recognize what is that based on the DNA sequence. We can also identify what is that, and also we can discover the new species. So that's how this became, this tool was introduced. Um, as I told you, being what exactly the need of the hour in order to explore the 10 million species. Okay, so people were thinking this is DNA based, and we all know how much of progress is being made in genomics. So genomics is going on. Technology is increasing day by day, where it has become a very, very high throughput system. So that means it can go on for 24/7. Okay, so that is the reason why I said I use the word high throughput. So once it is DNA based, then it can be automated as much as possible because so much advancement is happening. Okay, so the idea behind this is we can go for very speedy discovery of the unknown species in the earth, in the universe. Okay, when any technique comes, I think we will have because always it will be debated out what is the advantage and what is the disadvantage of that. If you see here on the advantages, as I told you, it will give ID. Of course, it is not exactly naming as in the taxonomies, but it will give some kind of ID. Just like in GenBank, we have accession number. Similarly, we have some kind of accession number quickly for all the species, which will be unique accession number. And also it will help people were saying the advantages ID and then discover new species so that we can fill those squares whichever is left. And also the speed is the one which is very, very essential in this time, because as I told you, as much as time we delay, we are going to lose as much of a species because of very, very quick urbanization. Okay, and also the finally once these things happen quite very fast, because as I told you, it's a high throughput technology. Then the collection is going to become a very, very big collection, because whatever we have right now, if it is in millions, it is going to be in millions or billions. So you can imagine the ecologist or whoever is involved with the species, they are going to have a big collection in order to work with. So that is some kind of a um, fully revitalizing the whole biological collection. Okay, so this is a big advantage for all the researchers. Okay, so these are all some of the big advantages which has come, and that's why it has become so popular now. And when it came, people also were opposing. They said it won't work because they said it may not be unique sequence and it might not identify a very specific species. And also they felt it will destroy the traditional systematics. That is, whatever it's already known taxonomically, they said it will destroy that because it is nothing to do with the morphology or anything. Instead, it is using only the DNA sequence. They said it may not work for all the species. Okay, so these are all some of the debates which are going on. And also they felt it is going to become a very commercial industry because it is becoming, any industry can involve and they can do all the sequences and then they can keep it, or they can just keep it for their own private use. Okay, so all this are happening, but still it has also advantage. And the whole thing, they named it as pseudo taxonomy. It's not a real taxonomy. So all this are plus and minus of this new technique. Okay, what exactly it is DNA barcoding? Before we go into exact technique, as the name suggests, as I told you, it involves some kind of a molecular biology because it's DNA, and it involves something kind of electronics where we need a scanner, which we see in the supermarket, to scan the barcode in order to know what exactly the product. So it is a kind of a molecular biology, electronics. So if you combine these two, then we can do the barcoding, DNA barcoding, high throughput barcoding. Okay, so it is not something new, which we already know what is the sequence. We already have the electronics for scanner. So the only thing is we are using it for the DNA sequence. Okay, that is the only difference. Otherwise, it's not something very new. Okay, coming back to the advantage as such, just to repeat, this technique, people feel, or the researchers feel, it is a very quick, cheap also, they say. Of course, in India, 10 years back when we started, it used to be like one reaction for sequencing. It used to be costing around 900 rupees or something like that, but now it has come to 350 or 400. So it is coming down every day. That is the reason why they say cheaper and cheaper. And I think for sure it will become just like maybe in 100 or less than 100 rupees per space. I mean per sequence sequencing reaction. So basically, it is quick and cheap. That's the reason why we call it as high throughput technology. And once it is there, if every lab does that, then there will be a speed discovery of millions of species yet to be named, so that we can fill those squares very quickly. And it will also provide vital new tools for appreciating and managing the Earth's immense and changing biodiversity. So this is something very, very interesting, because the field ecologist who is working in the forest and other places, now they have to spend so much time in order to find out the diversity by manually, or by going through and using different techniques, which takes many sometimes months and years. Okay, using this technology we can make these things faster. And other thing, very interesting thing about this technology is we can also study the changing biodiversity. So that means you can take a forest region and you can say what are all the species present today, and then in another year or 2 years, again we can do the DNA barcoding and say what are all the species. So maybe within 1 or 2 months you can finish all those things, because once the sample comes to the lab, because of a high throughput technology, this will be very quickly processed and we can name the species. So you can compare quickly what is the change exactly which has happened in the forest region in this very short time. So that will give a that will give a very, very significant information to the field ecology. So it is not only used for molecular biologists, it's also used for very classical ecologists, this information. So these are all some of the big advantages barcoding has. Okay, so initially it was it was used only for to understand the ecosystem and other things, to name the species, ecosystem and all. But slowly what happened was people started using this for many other commercial applications. For example, authentication of medicinal plants and products. So this is also one area which we are working, and food quality control. So um, these two, I think if I give an example, maybe you can better appreciate the things. Say in the medicinal plant, if you see, I think especially in this scenario, COVID situation, we all take many herbal related products. Things which we think it is giving some kind of a immune boosters or immunity. Okay, so they sell so many different brands in the shops. We just buy that and then we use it, but nobody knows exactly what kind of regulatory aspects it has gone through, and how authenticated the products are, because it is not like it's a traditional medicine. So there is no rigorous regulatory approval for all this. Anybody, if they want to sell, they have to just undergo a very small regulatory process in order to come to the market, uh, which is not the case in the modern medicine. Okay, there is a very, very strict procedure for that. Okay, so in these circumstances, barcoding can help to authenticate what exactly the herbal products which are sold in the market, so that we can say whether the species which has been mentioned on the vial, whether it is the same inside also, whether there is any adulterated or something like that. So in that way, it is quite useful technique in order to safeguard the health. Similarly, in the food, I think I can give one example. I think we all can recollect sometime back in Chennai, there was a big debate whether it is a goat meat or dog meat, because morphology looked like a dog, so everybody thought somebody has adulterated with a dog meat instead of goat. Okay, so in this kind of situation, just we need maybe 1 or 2 days time in order to quickly say what kind of a meat based on the DNA sequence. So, okay, so any food or any biological thing, if it is not processed fully, we can take the DNA and do this, uh, use this technology in order to name what is the species which is involved in that, whether it's a plant, microbe, or animal, or anything. Okay, and biodiversity studies, just now we saw like what all the different aspects where in which the biodiversity is quite useful using the DNA barcoding. And other application aspect is identify the juveniles. Juveniles are nothing but young ones. Say many of the species, whether it's a plant or animal or fish especially, during it is young, you can't recognize what is the species. Okay, so we need to have some kind of a tool quickly to say this is the species which you're going to use for breeding or any purpose. Okay, so this is also again has been proved very vital for identifying the juveniles where you can't differentiate morphologically. And many times it is also associated with the sex, in not very commonly used, but there are reports which they have used. Barcoding can be used to uh, say whether it's a male or female. Maybe the regions are different. The nucleic acid regions they will select as a different one. And then finally, it is quite interesting to say now people have started using for forensic analysis this technology, because as we all know, as I told you, the genomics is getting, every day it is getting changed. Technologies keep changing. Even picograms of DNA or micrograms of DNA is enough to say or to amplify in PCR and to know what is the sequence. So in the crime site, if they can find any of the biological material, even if one leaf or half leaf or any part, you can extract the DNA and say what is the species in that. So that can serve as a clue for the crime. So a small thing can give a big clue in order to solve the crime. So in that way, I'm just giving an example. There may be many other biological things which may be related to the crime, which you can use and find out what exactly happened, which is similar to DNA fingerprinting. It's not the same, but it's a different. So just like fingerprinting, here also we can use DNA barcoding in order to know the species in the crime site. Okay, so these are all some of the big applications which is being used. Whatever the tool which has come just to identify the plant, biological samples, now it is being used for many different purposes. Okay, what is happening all around the world? Because this technique has become so popular, and as the technique involves, we need to enrich the database, because it should be all the countries' effort. Because if one country does all the barcoding and other country doesn't do, it is not going to be very useful, especially country like ours, where we have so much of biodiversity. So what happened is internationally they formed many of the barcode of life projects, that is consortia. So what they've done is they have all joined together and formed some kind of a consortium, so that they will work on very specific fungus or fish or mosquito or butterfly or European Council barcode of life. So big associations have been formed, and people have started working so that they don't have to repeat the same in different places. Ultimate aim is to barcode all the things whichever is there exist on the earth, so that you can explore many new things and put it in the database. So for future generation, we can conserve that and use it for different purpose. Here I have to say, India is very known in fish barcoding, because there are big institutes which are working in fish barcoding. What they've done is they have barcoded many of the fishes, freshwater and also mostly freshwater fishes, because we have so much of resources. So if you put the publication, many will come from India. So India is known internationally for fish barcode of life project. So many members are from India. We are working on plant, but we are a very small group working on plant. And we are also attending all these conferences and other things. So we are connected with others, but we are not in a big group to do many of the things, but a very small group. Okay, so how do we do this? As a question in my title, I put why, we have explained how. This is what the slide. Okay, so the name is something very fancy, DNA barcoding. So, but it doesn't actually, the experiment doesn't say that. If you see here, the steps, even in college, we can just do all these things, because in the practical, you will be isolating the DNA. If you have a PCR, I'm sure PCR will be present in the biotechnology lab. So you can use the DNA, whatever you isolate using the CTAB method in the practical session, use that and then go for a primer and amplify that and then send it. Anyway, we are sending the sequence, outsourcing the sequence to the company only. So you also can do that. Anybody can do that and get the sequence in a week's time. And you can use the blast in order to see what is the closest species, closest sequence which is coming out. Okay, so if you see the steps, it is as simple as just PCR. So first step is, when we say barcoding, it is not something very small numbers, because normally, as I told you, it's a high throughput technology. So people will take hundreds of samples at a time, and then labeling is quite important. That's why databasing means hundreds of samples, whatever you select, you have to label very carefully. And imaging is one of the thing where we don't do any more rigorous preserving the samples and all that. Instead, you keep the image, high resolution image, as a reference material, and then you take a tissue sample without damaging the species, very small, maybe 50 mg or 100 mg of sample is fine in order to do the DNA extraction. Okay, and then go for a PCR. I'll tell you what exactly primer and other things in the coming slides. And then do a PCR, check whether you got the expected band, and then sequencing reaction, which will be done by the company wherever you outsource it, and they will give back the sequence by email. You can edit and then analyze it, and then see what exactly you wanted from the sequence, see whether it matches. Okay, so it is not very difficult. Anybody can do it, and we are also helping many labs in this aspect. Okay, how all this have started, because it is interesting to know anything whichever comes first, how exactly people have started to thought about all this barcoding and other things. First, when it came, it was first proved in animals, not in plant. In animal, what they've used is cytochrome oxidase 1, that is a mitochondrial gene. So we know, like genome, mitochondrial genome is there, nuclear genome is there, chloroplast genome is there. So like that, in plant, we have chloroplast and nuclear mitochondria. In animal, mitochondrial genome is there. So initially what they've done is, they found the cytochrome oxidase 1 to be during the evolutionary stages. This gene has given some kind of significant differences in the sequence between the species. So people have found out that, and they thought, why not we use this region in order to amplify that and see whether it can resolve all the species. And luckily, they found in animal it works 100% perfect. Means if you take the total DNA, like in the previous slide, whatever I showed, and then use the specific primer for cytochrome oxidase, forward and reverse primer, amplify that, and then do the agarose gel, and then see whether you got the expected size, and then you send that for sequencing. Once they give back the sequence, you can do the analysis. So it was very simple, very straightforward for the animal species. So that's how it started, the whole DNA barcoding thing. Okay, so now I'll come back to the plant, because we are specializing in plant. I'll talk about it. But before that, I'll just finish this barcode. Once we have the sequence, what exactly we can do with that? There is something called a very dedicated database in University of Guelph in Canada, that is Barcode of Life database. It is free. Anybody can access, create account, and you can submit, and you can use that for generating the barcode, and also for using the database, just like GenBank. But it is very dedicated for barcode sequences. So this is developed and hosted by University of Guelph, Canada. And what they have in this database is, they have a workbench for barcode projects. So that means whoever is working on barcode, they can go there, you can create account, and you can share your things. People also, already people would have shared. You can just use all this information and then enrich the database as much as possible. So they have a workbench for most barcode projects, as I told you before, like mosquito, fish, plant, and all that. And other interesting thing is, they have a laboratory information management system for assembling the data. So that means once you have the sequence, you don't have to go many different places in order to do the bioinformatics analysis. It's a one-stop solution where the bold database, you can put the sequence, you can do whatever you want, and it will give, I'll show you in the next slide what exactly we can do with that. So it is quite easy. We don't have to be an expert in order to use this system. So basically, this is used for management as well as for analysis of nucleic acids, the barcode sequences. This database is basically identification system for matching unknowns to the reference record. So what it does is, whatever the sequence, unknown sequence or known sequence you have in hand, if you want to see whether anybody has published or reported this sequence, if you put the query sequence into the database, it will give the related sequences and say you can say whether you have that in the database already, somebody has reported from different places. And then now this has been also linked with the GenBank. Means GenBank is a very wide database, very common database for all the gene sequences. So anybody, any gene sequence you have, you can use the GenBank. But this one is a dedicated one for barcode, that is bold. But now these are interlinked. Okay, but the big difference between these two databases are, GenBank is uncurated database where nobody is checking the information what they put in the GenBank, because it's a very huge database. No resources to curate it. But bold is a very limited one because only barcode data is being deposited here. It is fully curated. You have to undergo a very rigorous submission process in order to submit your DNA barcode sequence. So these two are the big differences, but both are linked now for the advantage of the users. Okay, how exactly the bold database looks like? If you see on the left hand side, this is a analytical chain in the sense how the steps, whatever we explained there, you can see on the specimen where you collect the specimen. You don't have these are high resolution pictures which you can see. You have to take that. You don't have to damage the species as such. So you can just need one small portion of it. And whatever the data is available, you can deposit that, so that people start depositing. You will have more information finally. So tissue sample you can see, just take, and photographs as many as possible, and other data also is being linked in the database itself. And on the bottom, on the left hand side, you can see the extracted DNA amplified, and then it is going for a sequencing analysis. On the right hand side, whatever the sequence, you can see here the chromatogram on the right hand side, the bottom one. You can see here the chromatogram which comes from the company. So they also send the sequence. So using this sequence, you can create a database in this bold laboratory information management system, and it will do all the things. We don't have to do manually. This will do it. And once it does, you can see here, it gives all the information, because we need to give latitude and longitude from where the species is collected. Based on that, it exactly say from which habitat it has been collected and what is the image of the species, and also the species information whichever is given. So all this are there in one-stop solution in bold. Okay, so if you see here, this is how the barcode of the sequence has been converted into a barcode. You can see the ID also. And on the right hand side, automatically in the bold system, if you give this, it will make a phylogenetic tree. And you can see the query sequence in the red, like here. And rest of sequence, what are all the closely related one, which are there, you can see here. So we don't have to be a bioinformatic expert in order to do this. You have to just put the sequence. It will give this phylogenetic tree or rough estimate what is closely related, already known one. Okay, so you can see all the information will come, not only your information, what you are giving, even the other information, whatever has been deposited already. So this is quite useful, especially for the taxonomists. Okay, so this is how the bold works. And this is the latitude, longitude, which I said. Based on that, it will calculate from where the species has been collected. We have to give all this information when you want to submit the sequence. So then only it will accept for the submission. Unlike in the case of GenBank, where it is much easier to do it. Okay, so this is regarding the database as well as the technology, how we do this technique, barcoding technique. Okay, so this is regarding the animal. It is very straightforward. Now coming to the plant, because we are working on this. We are not lucky. Plant scientists are not very lucky, as in the case of animal scientists, because it is complicated here in the sense there is no single candidate till now for a barcoding of a plant species, because for one group, some of the barcode primers are good, so barcode region primers are good, and for some it is not working. Say for example, in plant we have 3 different genomes: chloroplast, mitochondria, nucleus. And nucleus is the one, ITS is the one traditionally known, and still it is popular. And people have tried with different regions from chloroplast, from mitochondria, and from nucleus. This is a review which has come long time back, where you can see for each one they've given what is advantage and disadvantages of this region. Say low variation and rapidly changing, high identification ability but poor PCR amplification for different regions, and also low variation and low identification ability for RPOB, and for RBCL, very common candidate from chloroplast, which is showing high variation. It is good for barcode, and identification ability is also good. So it is now commonly used one. And PSB, again, in plastid region, low variation and low identification ability, but for some of the groups it is work. So even till today, there is no single candidate for the plant to say that it is a universal primer, just unlike in the case of animal, where cytochrome oxidase 1 works. Okay, in plant, you can see cytochrome oxidase is not very powerful because it is showing very low variation, and also it is rapidly changing during the evolution process. So that is the reason in plant it was not very successful. The quite successful candidates are whatever we use in the lab here in Bharatiya University is ITS, RBCL, and sometime other things like RPO or MATK and other things. Okay, how do we select the region like primer? I think this is a very general PCR primer, technology. So for barcoding, criteria is the different species whichever you are comparing has to have some kind of a difference, and it should not be too much of difference also. So we need to select a region where primer can be at the end of the sequences. For example, in the left and right side, we need to have a primer. So that means here it has to be conserved, and in between it has to be showing some kind of a difference. For example, I've just put 3 different scenario just to explain you easier. If you see here in this first scenario where we are comparing 6 different species, where you have taken a primer, where sequence is completely conserved, good for PCR because it is fully similar, but uninformative because you need to have some kind of a difference between the species in order to differentiate the species. So this is not very good. That's why we put fail. But in the second scenario, if you take something like sequence shows no conservation and impossible for PCR, because here so much of differences and difficult to PCR. So that means you can't have a universal primer, but it is good as a barcode. But we need a universal primer in order, because we can't keep changing for each plant the primers. It is no use. So this is also not very good. The ideal situation will be the region where we have some kind of a balance between the first two. So sequence shows some, that is 70% conservation, and it's good for PCR as well as good for as a barcode. You can see here some of the colors it is changing, for example, here and here. So like that, you have small differences. So using these differences, you can differentiate the species between each of them. So this kind of a region we need to select in order to say it is a good barcode. And this is a challenge in the plant. They could not come out with one region where it is successful in all the groups of all the families of the plant. Okay, for some family, some candidates are good, and the same good candidate is not working for other family. So like that, but still people are working on that. It doesn't mean that no candidate, so we don't have to stop the things. Still experiments are going on to find out. Okay, I think very briefly I've talked about what exactly and how it is. I'll come back to what we are doing in the lab as such. I think it's almost like time. I'll finish in 5 minutes. So these are some of the publications which we have done along with the group which I've showed, the BOLD database, whoever has found. So Stephen G. Newmaster, he is one of the BOLD database, the institute which has been created. So they are the pioneers in this field. So what we have done is we got a opportunity for the student to send there for a training, and she went there, Divya, a PhD student, and she could work with them. And what they've done was they've taken some of the herbal product which is available in North America, that is US and Canada, and they've done a barcoding, and they found so much of adulteration and other things. You can see here in the abstract, threat to consumer safety and all, and it has become a big issue in Canada. So this has gone in a big journal as a joint publication. Okay, then what happened was, because the ultimate idea of sending student to other countries, come back and establish the things in India itself. So she came back, and then we wanted to test all the Indian products here, herbal products. So we got many of the products from different shops, and we just tested it, similar to whatever they've done. So this has been published in Drug Safety. Again, it is a good journal, in a big, widely known journal. Again, we found so much of adulteration in the Indian market also. But for some reason, it has not become a big issue here, unlike in the case of Canada or US. Okay, so basically, we have spent so much of time on analyzing the herbal product, even though we started with identification of the plant species, but now we have gone to the products. Okay, so I won't go in detail because it's already time. So these are all some of the findings where you can see, out of 100% testing, like say around like 50 or 60 products, I think 89 herbal products or 50 medicinal plants, 40% we have found it is authenticated. That means authentic, whatever they've listed in the label is. 60% we found it is not authentic. So we have to be very careful when we buy outside in the shops, the herbal related things, because there is no strict regulation. People are selling just like that. So it doesn't mean that whatever we take herbal, it is safe. We have to be very, very careful in selecting maybe some of the things, brands which have a strict quality control may be good. So, but unknown ones, we have to be very, very extra careful, because it causes so much of other health issues like kidney and all that failures. Okay, I won't go in detail because already time. Okay, I'll just finish with this. So this is one of the facility in Canada where you can see these are all PCR machines. It's all linked. When we say high throughput, the advantage is all these machines can work 24/7 and throughout the year. It's all automated, robotic controlled. So only place where the human interference is needed is once the sequence comes. To analyze the sequence, we need humans in order to recognize what kind of a sequence, what is the species, and all that analysis part. Otherwise, it goes on. So that's the reason why we say it is a high throughput technology, so that we can fill the squares, whatever the blank left in the initial slide, quickly can be identified, and all these things can be conserved for future use. Okay, so these are all some of the developments which are happening. The sequences are becoming smaller and smaller, tabletop, which used to be very big instruments. And this is the one which is quite interesting. They say it is going to be handled like a mobile phone. So this can connect to the GPS and BOLD database. You can know what is the species in a very quick time if you have the sequence when you're going for a field trip and other things. And this is the ultimate, where you can carry this device, very small device. If you keep the tissue, it can extract the DNA, PCR it, and then it can amplify it, and sequence will be there. It is connected to the BOLD database, and immediately in the field itself, maybe in 1 or 2 hours, you will know what is the species which you are coming across in the field visit. So these are all some of the advancement which is happening. I think this technology is being developed by Motorola company. I think soon we might have this in the market, which might be expensive initially, but later it'll be cheaper, because whole thing is PCR, sequencer, DNA extraction, everything is inside. It's in a very, very small scale. That's why I said genomics is becoming so much of advancement every day. So these kind of high throughput technology is quite useful for identifying or giving ID for all the species before we lose or it becomes extinct. Okay, so once we have this situation, this is what we need exactly. Imagine a world in which you can know the name of any animal, any plant, any fungus, any organism on the spot in an instant, anywhere on our planet. Once we achieve, reach this stage, I think those squares can be filled in no time. So that is the, ultimate aim of why this barcoding is becoming popular, and why this is being promoted all over the world, so that before we lose many of the natural resources, at least we can start conserving it, so that the precious genomic information can be used for our future generations. I think that's it. I have acknowledgements. Thanks to Bharatiya University for all the funding support. Of course, our collaborator in University of Gulf, where we are in constant touch. And herbal industries, because as Dr. Francis was saying, we are working closely with the industries in order to authenticate their samples. And I should thank all of them in order to fund and for supporting our research activities. This is our lab in Bharatiya University. I think I'm sure it is useful. And as I told you, anytime, you can contact me for anything. We are ready to help. Thank you very much for your patience listening. Thank you.
Thank you, sir. Thank you for your mind-blowing, exemplary talk on the DNA barcoding of medicinal plants.
Thank you.
So it's a time for the participants. Sir, we have received a lot of questions in our chat box.
You can select.
So, yeah. So we have selected only some to clarify with the resource person. So I will list out some of the questions posted by our participants.
Yeah, sure.
So this is a question from Raju Biswas.
Okay.
In different environmental conditions, drugs or many herbal products, they are losing their activities, or it can be converted into toxic compounds. Can we use this technology in this problem? He's asking about the barcoding. Can we use this barcoding system for this?
I think the participant who has asked this, I think he's already involved in some kind of things related to whatever we are talking today. For example, secondary metabolites or medicinal plant and all that, because it's a so very relevant question which he has asked. And already we are coming across all these things when we say barcoding. Barcoding, what I should say is, this is only will name the species or identify the species. So it won't say what is the secondary, active ingredients, what is there in the plant on it, because we are not doing anything related to the metabolites. We are just touching only the nucleic acids. Okay, so that is the reason why in Canadian group now they are coming out with a new perspective on this. They are coupling both active ingredients as well as barcoding into one database, because it will address the question what the participant has asked, because we need to know both, then only it is commercial it will be successful. So these are two different things, but now the concept is coming to couple these two things from different regions, whether there is any difference, and then make a database so that that database will be useful for commercial applications.
I hope Mr. Raju Biswas is satisfied with your answer. With this, I will move on to the next question. So it's a question from, again, the same person, Raju Biswas.
Okay.
In bacterial identification, we are using 16S sequencing, whereas in the plant, we are using 18S sequencing. Why we are going for barcoding sequencing?
Actually, again, it is very interesting. 16S, 18S, all this are, again, it is a kind of a barcoding only. Only thing is, the name whichever we use currently is something fancy, but the same technology is being used in bacteria for quite long, but we don't call it as a barcoding that. We just say molecular identification or something like that. Okay, so, but technology-wise, it's same. 16 and 18S, these are evolutionarily conserved in these two groups. That's why we use two different things. Otherwise, barcoding can be used there also, but it is not being done, even though bacteria, for many years, we are doing the same kind of technology.
The question from Jackson. Can we isolate DNA from all the preserved herbal products?
Preserved in the sense, what, any idea, like preserved, where is the situation arises for preserved?
Yeah, so it is in the form of a powdered material, or it is in the form of herbarium.
Okay, yeah. I can tell two things. there are many publications. Yes, because as I told you, like in India, whatever we want, just go to the field and collect it, fresh sample, because it is so much available in India. But in temperate countries, whatever they want, they have to go and depend only on the museums, herbariums, or where they have preserved the sample. Okay, so many reports which are coming from temperate countries are basically they are working on hundreds of samples from herbariums or preserved ones in the museums. So still they could successfully isolate it. Even very old samples, they could get the DNA, because so much of advancements which has happened in the isolation, amplification and all that. So it is possible from the preserved, unless until it is fully processed. Say for example, if somebody is boiling it, say in the herbal product process, in the preparation process, then DNA, we would have lost the DNA after 70 degrees heating. So there we don't have a chance. But otherwise in the preserved and other things, there are so many reports which are there.
Sir, the question from Mahalakshmi Mishra.
Okay.
Is there any database for endemic species? Her question is there any database for endemic species?
So it is a taxonomy general database, is it? Whether she is asking general database or barcode database?
Generally.
General, I think there are many. Like even in India, we have different things from Bangalore or even in Ayush and other places where they have a separate database for, say, Western Ghats and other things. But I'm not sure, because we don't look into all that. Maybe I think you might be knowing some of things, Dr. Francis.
Yeah, so many databases are
there. There are many.
One more question from Amar Singh.
Yeah.
Is there any role of DNA barcoding in ex situ and in situ conservation of plants?
Not directly, as I told you in the slides. Once we say identify or name the species, I think then comes the role. Like so, basically, what we are trying to say is, if you don't even identify or preserve it, or if you don't even identify, then there is no question of preservation or conservation. Okay, so that is the reason where we say barcoding is going to help to name the species quickly, so that we can use those species and conserve it, so that anytime a taxonomist can come and do all the morphological study or any study, or genomics person can take the genes from any region of the genome and use it for other purposes. Once we lose this, that's it. Like we can't create something once we lose it. So barcoding is not directly helpful, but it is the starting point in order to conserve, for conservation, whether it's in situ or ex situ.
So it's a question from Praveen Prasad. DNA technology, can we use this technology to know the genetic disorders of brooders?
Okay. I think basically the regions whichever we select as evolutionarily significant regions. So our aim here is not to know the disorders, because for that we need to know the responsible genes for that particular disorders and study that. Then based on the mutation you can, but it is the same kind of approach which we are using here, because once the sequence changes in a particular region, whichever region which we are talking here, ITS, RBCL and all that, it is kind of a mutation where you see from one species it is different, in another species in different. So based on that difference only we use, but it is not connected to any disorders. It is basically, it has been selected for knowing the differences between the species. So disorders, we need to go for some other regions in order to know the disorders.
Yeah. So finally, the question from again Mahalakshmi Mishra. Can you describe a new species without DNA barcoding?
Yeah, of course. Like taxonomists were doing, are doing all the millions of 1.7 million or 2 million has been described by taxonomists thoroughly, and we are enjoying that now. So barcoding is not going to replace the taxonomy. It is only a supporting tool where, as I told you, it can't give a taxonomical say order, like in the case of taxonomist, which they give like order, phylum, species, genus, family, and all that. So here we don't do all that. We just give some kind of a ID and keep it so that we have ID. Later, taxonomists can take their own time in order to explain the species, because we need taxonomists in order to cross verify what is the species which we are working. Then, because then only we can say, then we can name the species. If it is not in the database, we can only say it is a new species, it to be named or something. We can't name it because we don't have a morphological characters for that. So taxonomy is needed always, and barcoding is going to support them to make the process, to speed up the process.
This is the last question of this session, today's session. It's a basic question, I think, from Meritini status. Whether DNA barcoding done in yeast and fungus?
Yes, we ourselves have done for helping others. We have done for fungus, because botany department, somebody was studying mycorrhiza and other things. So we were helping them. Yes, we are doing algae also, we have done. And sometimes the region differs. So we just go by the literature, but it is being done in all the groups, like algae, fungi, and all that. It works.
Okay, so thank you so much for your excellent answers for all the questions posted by our participants. So once again, I thank you on behalf of our department and our college management and my own behalf. Thank you, sir. This is a time for proposing vote of thanks. For that, I call upon Dr. David Raja, the young and energetic researcher as well as a teacher, to propose vote of thanks. Welcome, Dr. David Raja, sir.
Thank you, sir. Good evening is reset spot after winning our one-day innings. A very pleasant good evening to all. It is a privilege and great joy for me to extend a vote of thanks in this majestic webinar on plant DNA barcoding, what, how, and why. First of all, I would like to thank and praise God Almighty to giving this great opportunity. Next, I would like to extend my sincere thanks to Reverend Father Rector, Reverend Father Secretary, and Reverend Father Principal for their constant. Thank you, Fathers. I wish to express my sincere thanks to Dr. R. Satish Kumar, Professor and Program Coordinator, Department of Biotechnology, Bharatiyar University, Coimbatore. Sir, you made this day a fruitful one with your wonderful presentation and raising the meaningful questions like what, how, and why in the field of plant DNA barcoding. I believe all our participants get crystal clear information and idea about plant DNA barcoding. Thank you so much for accepting our invitation to be here as a resource person. Thank you, sir.
Yeah, thank you. Thank you very much. It's always my pleasure. I mean, I would have loved to come to St. Joseph's, but due to this situation, I could not. But it is always a pleasure because Dr. Francis is always in touch with me. And I thank you also as a co-organizer. Thank you very much.
I also thank Dr. Sagai Satish, Dean of Biological Sciences, and Dr. Sendil Kumar, Head of the Department of Botany, for their wonderful support and guidance. I especially thank Dr. Kanike Raj, the technical team head, and for their wonderful arrangement throughout the program. Thank you, sir. I convey my sincere thanks to Dr. Francis Xavier, one of the main organizing secretary of this webinar, for his hard work and meticulous planning. I also express my deep regards and thanks to all the staff members of Life Science departments for their fullest participation. Thank you, sirs. Now, my special and hearty thanks to all the participants from various parts of India and abroad for your wonderful participation and response. Thank you, friends. Before concluding, let me express my sincere gratitude and appreciation to all the well-wishers of this webinar for the grand success. The greatest history book ever written is the one hidden in our DNA. Once again, thank you one and all.
Thank you, Dr. David Raja. Thank you for your wonderful vote of thanks. Big thank you to you.
Yes, sir. Thank you.
Yeah, this is the time for our participants. I would like to give some reminders to the participants. Hope you got the attendance as well as feedback link in the chat box. So please fill the same and submit the same to organizers. So your these certificates will be issued to the registered participants who attended the whole session and participated in online attendance and the feedback. So many of the participants, they are frequently sending the mail, sir, we didn't get a feedback form, we didn't get attendance form. So we will not send the attendance and feedback form to the emails. So you have to fill the same in the chat box only. Within 15 to 30 minutes, we will close the feedback and attendance form. Please take care of the filling of both the forms. Then only we will provide the certificates. So please provide the correct details in the feedback as well as in attendance form. Especially in the feedback form, many people, they are doing incorrect informations. So please take care of during filling the feedback form. So it will be reflected in your e-certificates. With this, I'd like to remind one more information to the participants. So tomorrow we will have one more session on ethnopharmacology and drug discovery. So this session will be chaired by Dr. Sitapati, postdoctoral researcher from Maryland University, USA. So for this, I invite you all, join with us tomorrow exactly at 5 p.m. So have a great day. Thank you all. Thank you so much. Thank you, sir. Thank you for your
Thank you.
wonderful
Baklanga.
work, sir. Thank you.
Okay,
Yes.
Thank you, sir. Thank you, sir. Thank you.
Bye.
Thank you, David.
Okay, sir.